Hello, good morning students. Today we'll study parthenogenesis. Parthenogenesis is virgin birth. The offsprings produced by parthenogenesis may be of any sex, male or female. Various factors like chemicals, radiations, physical factors, presence of bacteria can induce parthenogenesis in insects. Parthenogenesis helps to increase progeny when conditions are not suitable for sexual reproduction. Parthenogenesis is a special type of embryo formation from an egg without fertilization by a sperm. It is distinct from asexual reproduction as it involves the production of egg cells and there is no contribution of male gamete. Parthenogenetic development is affected by meiotic or mitotic divisions. Obligate parthenogenesis. There is no sexual reproduction. Examples include several aphid species and willow sawfly, that is nematus, which reproduce through sexual reproduction in its native holarctic habitat and through parthenogenesis when it has been introduced into southern hemisphere. Obligate parthenogenesis in this invasive species allows a population to multiply and invade more rapidly. Facultative parthenogenesis. Depending upon environmental condition, organism can show parthenogenesis or sexual reproduction. For example, facultative parthenogenesis like in mayflies. A female show facultative parthenogenesis if a male is absent from the habitat or it is unable to produce viable offspring. Haploid parthenogenesis. In haploid parthenogenesis, haploid egg produces haploid adults. For example, honey bee. In honeybee Apis mellifera, unfertilized egg normally develop into haploid males by Arenotochus parthenogenesis. They are known as drones. Unfertilized eggs are produced by queen for the production of male. Unmated queenless worker whose egg also produce functional male. Diploid parthenogenesis. In diploid parthenogenesis, there are two pathways. Number one, automixis. It is post-meiotic process in which a haploid cell may either duplicate its chromosome or fuse with another haploid cell to develop a diploid zygote and grow into a diploid adult. Such organisms are not true clones of the mother. For example, stick insects. The genetic composition of the offsprings depend upon the type of the automixis, that is restitutional meiosis of anaphase 1 and fusion of its products, that is the offsprings get all to more than half of mother genetic material and heterozygosity is mostly preserved. This is because in anaphase 1 the homologous chromosomes are separated. Restitutional meiosis of anaphase 2 and fusion of its product. A little more half the mother's genetic material is present in the offspring and the offsprings are mostly homozygous. This is because at anaphase 2 the sister chromatids are separated and whatever heterozygosity is present, it is due to the crossing over. Second type is apomixis. There is no meiosis and two genetically identical diploid egg cells are produced from a parent cell through mitosis. The offsprings are cloned. They are the clone of the mother, for example, aphids. Now see, the process of sexual reproduction versus several forms of parthenogenesis. In sexual reproduction, there is a meiosis. The meiosis, it leads to the formation of oocytes or spermatocytes, right, which forms egg cell or sperm cells. Both will fuse to form a diploid zygote and the diploid individual is formed. This is sexual reproduction. Now coming to haploid parthenogenesis. In haploid parthenogenesis, meiosis is there, oocyte is formed, which form haploid zygote, and the haploid individual is formed. This is there in haploid parthenogenesis. Now coming to diploid parthenogenesis. Here we have got two forms. One is automixis, another is apomixis. In automixis, meiosis is there, oocyte is formed, right? Chromosome replication is there, and the same they fuse, right? And the diploid zygote is formed, right? So the oocytes, they fuse with each other in automixis. Now coming to the apomixis. In apomixis, there is no meiosis, there is mitosis. Female egg cell which is diploid, right? And the diploid egg forms the diploid individual. So in this way, we can differentiate haploid parthenogenesis from diploid parthenogenesis. Now coming to the thelytoky. In thelytoky, the unfertilized eggs laid by the female insects produce only female progeny. For example, aphids and odontocym, that is the parasitoid hymenopterans. It is based on either mitosis, that is apomixis, or meiosis, that is automixis. Apomixis. The female offsprings are similar to the mother and are referred as clones because there is no meiosis leading to chromosomal rearrangement. Automixis, it involves meiosis, but diploidy is attained without fertilization. Example is scale insects, whitefly, leaf insects. Automixis occurs through one of the following ways. First is either duplication of genome before meiosis, creating a cell with 4n chromosome, and then meiosis. Second, fusion of two nuclei during the meiosis. Third, a haploid nuclei resulting from meiosis is duplicated by mitosis to fuse again with itself only. So in this way, these are formed. Next is arenotoky. In arenotoky, haploid males are produced from unfertilized egg laid by the mated female or by secondary or supplementary queens which have not been impregnated. Offsprings from the unfertilized egg consist only of male individual. Example, wasp, bees, ants and thrips. In honeybee, when unmated queens are stimulated to produce unfertilized egg by exposure to carbon dioxide, they produces only males. They can produce diploid female offsprings via thelytoky like queenless unmated workers. In contrast, when mated queens lay unfertilized egg, they produces males by arenotoky. This indicates an extraordinary ability of queen to manipulate the kind of parthenogenesis that occurs when they lay unfertilized egg. Thelytoky and arenotoky in unmated queens and arenotoky only in mated queens. It is also suggested that mated queens could potentially produce daughters both sexually and asexually. Then is deuterotoky. In deuterotoky, parthenogenesis, unmated females produce unfertilized egg forming both male and female. For example, pear sawfly, that is Calliora. Males are often found in the laboratory population of Thelytocus species, and their frequency usually depend upon the temperature at which their Thelytocus mothers develop heterogony or cyclic parthenogenesis. Some aphids and water fleas undergo cyclic parthenogenesis. In these species, generation of offspring produced from fertilized egg may alternate with those produced from unfertilized one. Cyclic parthenogenesis may be caused due to number 1, seasonal temperature changes, number 2, eggs produced through sexual reproduction, have a greater ability to withstand the winter cold. They lie dormant until temperature rises. In Chalcids, the first generation, consisting of males and female, produces a second generation of female only. These female lay unfertilized eggs that produce both male and female, thus repeat the first generation. Pseudogamy or gynogenesis or sperm-dependent parthenogenesis. Pseudogamy is shown in life cycle of few insects like bark beetle, where sperms are only to stimulate the initial development of egg, and the sperm's genetic material is not used. Parthenogenesis only during gall forming stage. Gall forming aphids like pemphigus, wingless females produce parthenogenetically during the gall forming phase of their life cycle, and winged insects, when emerge from gall, reproduce sexually. Geographical parthenogenesis. Monosexed species with population of only one sex in insects occur in isolated environments such as island or high altitude region. For example, grasshopper, a diploid hybrid between two undescribed sexual species, referred to as P169, maternal sexual ancestor, and P196, paternal sexual ancestor, both restricted to small arid area of southwestern Australia. Parthenogenic population tend to occur at higher altitude and higher elevation at xeric, that is dry conditions, generally in more marginal and disturbed environment. They adapt poorly to rapid environmental changes due to low or zero genetic variability among their offsprings. Parthenogens predominate in marginal habitats simply because they are better colonizer. Single female is sufficient to found a new population, increase faster by foregoing the cost of producing male. Next is thycoparthenogenesis. It is the ability of spontaneously generate female offspring from unfertilized egg of sexual species accidentally, and haploid gamete become diploid gradually during development. For example, Locusta migratoria. Next is induced parthenogenesis by bacteria. Endosymbiotic bacterium Wolbachia modifies host reproduction in order to enhance its own spread through the induction of parthenogenesis, where males which are unable to transmit Wolbachia are not produced. Thalitoki is induced by the presence of bacteria Wolbachia in Loxoblemus, the only species of Garralidae family to show parthenogenesis. In Hymenoptera, the mechanism of diploidization due to Wolbachia infection, known as gamete duplication, is a post-meiotic modification. During gamete duplication, the meiotic mechanism is normal, but in the first mitosis, the anaphase is aborted. The two haploid set of chromosomes do not separate, thus result in single nucleus containing two identical sets of haploid chromosomes. Next is parthenogenesis in Phasmatodea. Rare fertile males could be collected in a genus Timea endemic to California, and their presence is estimated to be less than 0.2% of the population. The sexual determination of Phasmatodea is based on a system where the individual carrying a pair of autosomes XX are females and those with only one X, that is XO, are males. Male develop by a spontaneous loss of X chromosome during oogenesis. Advantages of parthenogenesis. It ensures the reproduction of organism when conditions are not favorable for sexual reproduction. It is advantageous to organisms that must remain in a particular environment and in places where mates are scarce. Numerous offsprings can be produced by saving a great amount of energy when the population density is low in case of extreme habitat and finding a sexual partner is difficult. Disadvantages of parthenogenesis: lack of genetic variation as there is no movement of genes from one population to another. Thank you www.google.com for providing us beautiful pictures.